Assalamu alaikum everyone. In this video lecture, we will be discussing few MCQs of cell membrane and transport, which is the topic of AS level biology 9700. Let's start with MCQ number 1. Which transport mechanism does not require a concentration gradient to be present in order to take place? So guys, what is actually meant by requiring a concentration gradient? A process requires a concentration gradient when the molecules have to move randomly from a higher concentration to lower concentration. So passive processes like diffusion and osmosis, they require a concentration gradient because molecules have to move from a higher concentration to lower concentration, right? So over here, facilitated diffusion requires a concentration gradient, osmosis requires a concentration gradient, transpiration also involves diffusion of water vapor from a higher concentration to lower concentration from the leaf to the atmosphere. Exocytosis is a process which does not require a concentration gradient. Exocytosis will involve the movement of very large molecules out of the cell irrespective of the concentrations. So the answer for MCQ number 1 is A. Exocytosis is an active process. It does not require a concentration gradient to occur. Let's move on to second MCQ. MCQ number 2. High concentrations of ethanol can disrupt cell membrane structure by denaturing proteins and increasing the separation of adjacent phospholipid molecules. As a result, cell membranes can decrease in thickness by up to 30% and become more permeable. So guys, they're saying that ethanol can disrupt the cell membrane by denaturation of proteins and by the separation of adjacent phospholipid molecules. They have also stated that the separation of the adjacent phospholipid molecules causes a decrease in the thickness of the membrane up to 30%. So guys, this could be the picture that normally what happens is that we have a phospholipid bilayer, right? This is the phospholipid bilayer, and the phospholipid molecules are not separated, they are close to each other, right? So what could happen in the presence of ethanol is that the phospholipids separate, and when they separate, what happens that the phospholipid in the lower layer, or we can say the tails of the phospholipid in one layer or lower layer, can the tails of the phospholipid in the lower layer can slide along the tails of the phospholipid in the upper layer, right? So this can give a picture somewhat like this that I'm drawing right now, and this will result in the decrease in thickness of the membrane, right? So we can see that phospholipids are separated and the tails of the phospholipids in one layer slide over the tails of the phospholipid in the other layer, and this results in decreasing the thickness of the membrane, right? This what happens when ethanol is present, and this is how ethanol disrupts the membrane structure. So it's written over here that yeast cells release ethanol as a waste product of of metabolism. So basically yeast cells, they carry out anaerobic respiration and they produce ethanol. So they release ethanol as a waste product of metabolism. They're releasing ethanol out of the cell. In response to increased ethanol concentration in their environment, yeast cells are able to increase the tolerance of their cell membranes to ethanol. So basically they are saying that yeast cells have the ability to tolerate high concentrations of ethanol, maybe because of some changes in their membrane. So which statement correctly explains a response to ethanol and could account for the increase in the tolerance of yeast cell membranes to ethanol? So guys, we have to select a statement which could account for the increase in tolerance of the yeast cell membranes to ethanol. Let's look at option A, decreasing the ratio of saturated fatty acids to unsaturated fatty acids, which means that there is a decrease in the saturated fatty acids and increase in unsaturated fatty acids, right? The meaning of decreasing the ratio of saturated fatty acids to unsaturated fatty acid means that saturated fatty acids are decreased and unsaturated fatty acids are increased within the cell membranes. This helps to prevent the tails of phospholipids on one side of the bilayer from sliding past the tails of the phospholipids on the other side of the bilayer. Guys, this could be true because if the tails are more tails are unsaturated, so they are less likely to slide, right? They are less likely to slide past each other. Do you get my point or no? If tails are unsaturated, so they will be kinky, and it's difficult for them to slide past one another. For example, if this tail is kinky, or we can say if this tail is kinky, it will not be able to slide over this tail, right? And this will not reduce the thickness of the membrane and the permeability of the membrane, right? So we can say that this could be an option. This could be a true option. Why? Because if the yeast cell decreases the ratio of saturated fatty acids to unsaturated fatty acids, more unsaturated fatty acids will be there, and as you all know that unsaturated fatty acids have kinks, right? And they are less likely to slide over each other. So phospholipids in one layer, the tails of the phospholipids in one layer will not slide across or past the tails of the phospholipids in another layer, and this will prevent the decrease in the thickness of the phospholipid bilayer, and also this will not increase the permeability, right? So answer is A, right? Let's move on to MCQ number 3. In an investigation, a plant cell was placed in pure water. The initial rate at which water molecules entered the cell R was greater than the initial rate at which the molecules left the cell. So guys, they are saying that plant cell is placed in pure water. This is the plant cell, and this plant cell is placed in pure water. And they are saying that water enters the cell. Obviously, water will enter the cell. So they are saying that the rate of movement of water into the cell, that is R, is much greater than the rate of water movement out of the cell. And this is true because water potential is higher water potential of pure water is higher, and the water potential of the cell is lower, obviously. So obviously more water is entering into the plant cell than the volume of water that is coming out, and obviously this rate R is greater than the rate of water flow out of the cell, right? In a second investigation, a plant cell of the same type was placed in a solution with a water potential equal to that of the cell contents. So guys, if this plant cell is placed in a solution that has water potential equal to, if the same plant cell is placed in a solution that has water potential equal to the cell, so the rate of water loss will be equal to the rate of water uptake, right? So there will be no net movement of water into or out of the cell, and obviously the rate of movement of water into this cell will be lower than R, and R was the rate when water was moving into the cell when the cell was placed in pure water. So what will happen in the second investigation over a period of 5 minutes? Water molecules will enter and leave the cell in equal amounts. This is true, both at an initial rate that is less than R in the first investigation. Exactly. The rate of uptake of water in the cell when placed in a solution of equal water potential will be lower than the rate of water uptake into the cell when the cell was placed in pure water. So if we compare the rate with R in the second investigation, the rate will be lower. The rate of uptake and the rate of water loss will both rates will be lower than R in second investigation, The answer is B. Let's move on to MCQ number 4. The diagram shows a liposome. Liposomes can be used to move therapeutic drugs into cells of the body to treat conditions such as cancer. Which row shows the property of a drug that could be transported in the sections of the liposome labeled 1 and 2? So guys, if a drug has to be transported using a liposome into a cell, and if the drug has to be placed in this location, at one location, so location 1 is basically the hydrophobic region, and the drug should be hydrophobic to be carried out in location 1, right? And the drug has to be hydrophilic if it has to be in the region 2. Why is that so? Because this region over here is surrounded by the heads of the phospholipid, right? And this region is hydrophilic, right? And this region is hydrophobic, right? So the answer is C. MCQ number 5. The indicator cresol red changes from red to yellow when put into acid. 4 blocks of agar containing cresol red were to cut to different sizes measured in millimeters. The blocks were submerged in acid. All other variables were kept constant. The time taken for each of the blocks to completely turn yellow was recorded. Which of the 4 blocks became completely yellow most quickly? So guys, in this question they are saying that there's an indicator called cresol red which changes from red to yellow when put into acid, right? So there were 4 blocks of agar which contained cresol red, and they were cut into different sizes measured in millimeters, right? And then these blocks were placed in acid. Now the time taken for the color to turn yellow was recorded. So they are saying that which of the 4 blocks became completely yellow most quickly. So if the block has to become completely yellow most quickly, so this means that the rate of diffusion must be the fastest, right? Rate of diffusion of what? The rate of diffusion of acid into the block should be fastest, right? And what do we keep in mind when we are looking for the rate of diffusion in any block? Just look for the smallest dimension. So guys, which of the option out of 4, or which of the 4 blocks has the lowest or the least dimension? That is A, with the dimension of 3. So the answer is A. If any block has the smallest dimension, the rate of diffusion will be fastest in that block. This is the rule. The answer is A. Let's move on to MCQ number 6. Diagram 1 and diagram 2 show how the transfer section through a leaf changes when the leaf is moved from solution X to solution Y. Which row describes the water potential of the leaf cells and the surrounding solutions in diagram 2 compared with diagram 1? So guys, when the leaf was placed in solution X, the leaf became flaccid, right? It means that the water left the leaf by osmosis from a higher water potential to lower water potential. Do you get this point? When the leaf is placed in solution X, the leaf lost water and the leaf turned flaccid, right? It means that solution X has a very low water potential, right? When the leaf was placed in solution Y, the leaf became turgid because the water entered the leaf cells by osmosis from the solution. So we need to compare the solution X and solution Y. We also have to compare the water potential of leaf cells. So what is the water potential of leaf cells in diagram 2 compared with the water potential of the leaf cells in diagram 1? So guys, in diagram 2, leaf is turgid. So leaf cells have a higher water potential as compared to the leaf cells in the diagram 1. In diagram 1, leaf cells are flaccid. So guys, water potential of the leaf cells in diagram 2 is less negative or higher because these cells are turgid. And in diagram 1, the leaf is flaccid with the cells having a lower water potential or more negative water potential, right? So leaf cells in diagram 2 have a less negative water potential as compared to the leaf cells in diagram 1. If we talk about water potential of solution Y as compared to the water potential of solution X, so guys, we saw that solution Y has a very high water potential, and this is true because the water is entering into the leaf from solution Y, making the leaf turgid. So solution Y has a higher water potential or less negative water potential as compared to solution X. The answer is A. Let's move on to MCQ number 7. The diagram shows a phospholipid molecule divided into three regions X, Y, and Z. R in region X represents a range of possible chemical groups. Okay. Regions X, Y, and Z affect the properties of cell surface membrane in different ways. Which row shows the effect of each region on the property of cell surface membrane? Okay, guys. So, what is the property of X? X is basically a hydrophilic part of the phospholipid. This molecule is the phospholipid, and X is the hydrophilic part. So, it attracts water molecules because X is the hydrophilic part. Repels polar molecules. These hydrophobic tails, both Y and Z, they repel polar molecules, right? And which of the part increases the permeability of hydrophobic region? So guys, always remember this point, that permeability of hydrophobic region is increased by unsaturated fatty acid, right? So over here we can see there's a double bond, there's a kink, and the unsaturated fatty acids, what they do, that they increase the fluidity and they increase the permeability. So it's Z which increases the permeability of hydrophobic region. The answer is D. Let's move on to MCQ number 8. When living pancreatic cells were placed in a solution of red stain called neutral red, the cytoplasm became red. The cells were then removed from the solution of neutral red. Okay. The red stain in the cytoplasm moved into vesicles which were exported from the cell, eventually leaving the cell colorless. Which transport mechanisms could explain how the red stain entered and left the cells? So guys, they're saying that initially the pancreatic cells were placed in a solution of a red dye or a red stain called neutral red, and the stain moved into the cells and the cytoplasm became red. Then the cells were removed from the neutral red and eventually the cell became colorless. How? That the stain that was in the cytoplasm, it moved into the vesicles, and vesicles were exported out of the cell, right? So they are simply saying that which transport mechanisms could explain how the red stain entered and left the cell. So guys, most probably the red stain has entered the cell by diffusion, right? It cannot enter by osmosis, right? It could enter by diffusion, right? Or maybe facilitated diffusion, and it exited out of the cell, or it moved out of the cell by exocytosis because it was in the vesicles, right? And vesicles are transported out of the cell by exocytosis and not by endocytosis. So the answer will be B. Active transport and facilitated diffusion could not be the answer because the stain was in the vesicles. It could not come out of the cell by facilitated diffusion, right? So the answer is B. MCQ number 9. A student wrote 3 statements about cell signaling, right? Which statements are correct? A signal chemical always has the same shape as the protein receptor on the target cell. Guys, this is a wrong statement because the signal chemical has the complementary shape, not same shape. What is meant by this statement? For example, if this is the protein receptor, so the signaling molecule will be like this. They don't have protein receptor and signaling molecule, they don't have the same shape, they have the complementary shape. So point 1 is wrong. An increase in temperature may decrease the effect of cell to cell signaling. Guys, this could be true because when we increase temperature, proteins can denature and they could decrease the effect of cell signaling. A mutation may decrease the production of active protein receptors for the cell surface membrane. This is true as well because a mutation in a gene coding for the protein receptors could lead to a decrease in the production of protein receptors. So the answer is D. Let's move on to MCQ number 10. A student set up an experiment to investigate diffusion. A block of agar 1 cm by 1 cm by 1 cm was stained uniformly with a water soluble blue dye. The block of the agar was put into a test tube containing 10 cm cube of distilled water at 20 degrees Celsius. The intensity of the blue color of water after 5 minutes was measured. So guys, what they are doing in this experiment, is that they are investigating diffusion, and for that they took a block of agar jelly that was 1 centimeter by 1 centimeter by 1 centimeter. It was stained uniformly with a water soluble blue dye, and then this block was placed in a 10 centimeter cube of distilled water at 20 degrees Celsius. So what happened is that the blue dye, because the blue dye was soluble in water, so it diffuses out of the block into the water, giving water a blue color, right? And the intensity of the blue color of water was then measured. So obviously, the greater the blue dye that comes out of the block into the water, the greater the intensity of blue color of water. Do you get my point? So the greater the dye that comes out of the block into the water, the greater the intensity of the blue color of water. So they are saying that 4 other experiments A, B, C and D were then carried out using different numbers of agar blocks, different sizes of agar blocks and different temperatures. All other variables were standardized. Which experiment would give a lighter blue color in the water after 5 minutes compared to the first experiment? So guys, they are saying that which of the options will give a lighter blue color in water after 5 minutes compared to the first experiment. Guys, lighter blue color means less diffusion of blue dye into water, right? And basically there are sizes of blocks and number of blocks. So you should know that for a lighter blue color, less dye should come out of the block into the water, right? As compared to the first experiment in which the block was 1 and it was 1 centimeter by 1 centimeter by 1 centimeter. So guys, we need to take a block which is smallest of all. First of all, we need to choose a temperature that is equal to 20, because when temperature is increased, the rate of diffusion is faster, right? So we need a slower rate of diffusion comparatively. And what else do we need? We need smaller blocks. Why? Because smaller blocks will contain less blue dye, and less blue dye will diffuse out. So the intensity of the water outside the block, so intensity of blue color in the water outside the block will be low if the block contains less blue dye. So if the blocks are smaller, like in A and B, and if the blocks are less in number, so there will be less total dye that has to come into the water by diffusing, right? So the color will be lighter blue as compared to the first experiment. So in the first experiment, the block was larger, 1 centimeter by 1 centimeter by 1 centimeter, right? And in this case, the block is smaller, 0.5 by 0.5 by 0.5. And in this option, we have 2 blocks, whereas in option A we had 8 blocks. So this would give a darker blue color. So we will choose B, because there are less number of blocks, which means less dye that can go into the water and give it blue color, right? So the intensity of the blue color in the water will be least, or there will be lighter blue color in water if the block is 0.5 by 0.5 by 0.5 and there are 2 blocks and temperature is less, 20 degrees Celsius, because rate of diffusion will be slower. So the answer is B. Okay. Moreover, we can also do one thing, is that we can also justify it by calculating the volume. So guys, what is the volume of this block? 1 by 1 by 1. So length into breadth into height, volume is 1 centimeter cube. If we find the volume of this cube, the volume will be 0.5 into 0.5 into 0.5. That will be 0.125, and if we multiply it by 2, the answer is 0.25. So guys, the volume, overall volume of these 2 blocks will be 0.25 centimeter cube, and the volume of this is 1 centimeter cube. So the volume is less for the blocks, right? And they contain less dye, so less dye will diffuse out, and there will be a lighter blue color in the water. So I hope you get it now. MCQ number 11, which description of cell surface membrane permeability is correct? Okay, which description of the cell surface membrane permeability is correct? An increase in the concentration of cholesterol molecules in the cell surface membrane can increase its permeability to hydrophilic substances. No, guys, if you increase cholesterol in a membrane, this will decrease the permeability to hydrophilic substances because cholesterol molecules have hydrophobic tails and they prevent the passage of hydrophilic or polar substances. Cell surface membrane permeability to large hydrophilic molecules is high. No, it's not high, it's low, and can be increased by membrane transport proteins involved in facilitated diffusion. Guys, the permeability to large hydrophilic molecules can be increased by membrane transport proteins, but it's not normally high. So B is wrong. The permeability of the cell surface membrane to ions increases as the proportion of saturated fatty acid chains in the phospholipids increases. No, guys, saturated fatty acids or unsaturated, they both make they both prevent the passage of ions through the phospholipid bilayer. Without the presence of carrier and channel proteins, the cell surface membrane has low permeability to large polar molecules. This is true, guys, that without the presence of the transport proteins like carrier proteins and channel proteins, the cell surface membrane is less permeable to large polar molecules. MCQ number 12, a plant cell with a water potential of minus 600 kPa was placed in a solution with a water potential of minus 410 kPa for 10 minutes. Which row is correct? Okay, guys, so they're saying that there's a plant cell which has a water potential of minus 600, and this was placed in a solution which has a water potential of minus 410. So what will happen? So we know that water flows from a less negative water potential to more negative water potential. So guys, water will move from the solution into the plant cell. That is because solution has a less negative or higher water potential and cell has a more negative or lower water potential. So water will enter into the cell by osmosis. So there is net movement of water into the cell, and water potential of the cell obviously becomes higher when the water enters the cell. Obviously, if water is entering into a cell, water potential of the cell will become higher and the cell will become turgid. Answer is A. Let's move on to MCQ number 13. A decrease in some factors will increase the fluidity of the cell surface membrane. Which pair of factors, when decreased, will increase the fluidity of the cell surface membrane? So guys, we have to select some factors which, when they will decrease, they will increase the fluidity of the cell surface membrane. So let's start selecting. So when the distance between the phospholipid molecules decreases, the fluidity does not increase, right? Always remember this point. Whenever phospholipid molecules are far apart, fluidity is high, and when they are close, fluidity is low. So if the distance between phospholipid molecules decreases, they will become closer, and the fluidity will decrease, not increase, right? We have to select an option in which the fluidity will increase by decrease in a factor, right? Proportion of short fatty acid chains. So guys, if we decrease the proportion of short fatty acid chains, it means we are putting up long fatty acids. Long fatty acids, what they will do, they will further decrease the fluidity and not increase it. Distance between phospholipid molecules is decreased. No. Temperature is decreased. No. Decrease in temperature will decrease the fluidity, and decrease in the distance between the phospholipid molecules will also decrease the fluidity. Proportion of phospholipids with saturated fatty acid chains. If we decrease the proportion of phospholipids with saturated fatty acid chains, this will increase the fluidity. Why? Because the saturated fatty acids, what they do, that they normally decrease the fluidity. So if you decrease the saturated fatty acids, it means you're increasing unsaturated fatty acids, and this will increase the fluidity. So this is true, because we need to decrease a factor that increases the fluidity, right? So if you decrease the proportion of phospholipids with saturated fatty acid chains, it means you're increasing the proportion of phospholipids with unsaturated fatty acids, and that will increase the fluidity. Proportion of long chain fatty acids. If you decrease the proportion of long chain fatty acids, it means you're putting up short chain fatty acids, and this will increase the fluidity of the membrane. Guys, always remember this point, that longer the fatty acid tails, lower the fluidity. Shorter the fatty acid chains, higher the fluidity. right? So C is the answer, because if you decrease these two factors, this will increase the fluidity of the membrane. MCQ number 14. The diagram shows a cell surface membrane. Which statements about the labeled molecules in the membrane are correct? Right. One is involved in the diffusion of ions. So guys, this is wrong. What we need to do is that we need to state that which of the statements for the labeled molecules is correct. So one is the phospholipid, and phospholipid is not involved in the diffusion of ions, right? If we talk about 2 is involved in facilitated diffusion. Yes guys, it's true, because 2 is a transport protein, which is a channel protein basically, and this is involved in facilitated diffusion. That's correct. If we talk about 3 is involved in the recognition of antigens. Guys, 3x is antigen. 3x is antigen, and it's not involved in the recognition of antigen, right? This is wrong. 3x is antigen, not involved in the recognition of antigen. For recognizing antigens, there are antibodies or there are receptors, right, which recognize the antigens. So this is wrong. 4 is involved in membrane fluidity. Yes guys, 4 is cholesterol, and definitely 4 is involved in membrane fluidity. So the answer is 2 and 4. MCQ number 15. Equal sized potato pieces were placed into test tubes containing equal volumes of different concentrations of sucrose solution and left for 30 minutes. All other variables were controlled. After 30 minutes, the potato piece in one of the concentrations of sucrose solution had not changed in size. What can be concluded from this result? So guys, they are saying that there are equal sized potato pieces and they are placed into the test tubes containing equal volumes of different concentrations of sucrose solutions. So guys, different potato pieces are placed in different sucrose solutions of different concentrations, and each potato piece has an equal size, right? And they are kept for 30 minutes, right? So there is one potato piece in one of the solutions of sucrose which did not change in size. It means that there is no net movement of water. So what can be concluded from this result? There is no net movement of water into or out of the potato. This is true because the potato does not change in size. It means that there is no net movement of potato into or out of the potato, right? There is no net movement of water into or out of the potato. The water potential of the potato is same as the water potential of sucrose solution. This is also true because if a potato is placed in a solution and there is no change in the size of the potato, this means that there is no net movement of water into or out of the potato, and that's because the water potential of the potato is same as the water potential of the sucrose solution. The concentration of sucrose in the potato is same as the concentration of sucrose solution. Guys, this is wrong. Concentrations cannot be same. Even though water potentials are same, concentrations are also same. But concentration of sucrose will not be same. Let me tell you why. Because inside the potato there is cytoplasm, there are other solutes as well. So the total concentration of the potato solution will be same as the total concentration of the sucrose solution, but the concentration of sucrose in potato and solution will not be equal, right? Because there are other solutes as well in the potato and not only sucrose. So the answer will be 1 and 2 only. The answer is B. Okay guys, so we are done with the MCQs. Thank you so much for watching. Don't forget to subscribe to my YouTube channel. Allah Hafiz.